Okay, thank you. So, because this audience is really very diverse, thought I'll do two things. First, there aren't too many slides. What I hope to do is manage or engage with an interaction between all of you. Uh, of course, you can ask any question at any point of time, but I'm also going to keep throwing some questions at you. We'll see how that goes. Um, the, the, the second part, what I want to say is, so please be as engaged and as involved as possible. Any kind of doubts, we'll try to clear. The second part, what I'm going to try to do is not get into too many details of either metabolism or networks or any such thing. Hopefully, you will get a feel for that as we go along in this program. Can you all hear me, by the way? Okay, thank you. Some volume control, good, because I tend to speak softly. The other thing I want you guys to get some feeling of is to reduce the abstraction. Forget about the equations and so on, but only think about what a cell or a tissue is trying to achieve in biology. And that comes down to understanding the flow of metabolism. Metabolism is not a static entity with one reaction going on. And because of that, if you can start to think of flow directions of reactions and therefore where it would end, that's a useful way to think about metabolism. At least I think that's a useful way to think about it. Okay, so just putting stuff down on a slide. Um, Thanks to Jonathan. It was a really useful, I think, introduction primer. All the little molecules of the cells were covered. This need for energy was covered. All this basic stuff was covered. So I don't have to do that. I don't have to build up a cell. So we will skip that part completely. But a lot of it is about what are the different molecules that make up a cell. Also done. Done. How much? Especially in the later half of the first talk, you heard about approximate numbers of molecules that are there in cells. So we are done with that. The other part we'll touch upon today, not going too deeply, is how much energy a cell needs versus how much a cell can make. Okay? And this difference usually decides what the fate of a cell is. We'll try to get to how to think of complete biochemical networks, but before that, we have to appreciate the individual entities of any network. And then we'll try to touch upon what are important in a whole network. If you're looking at a series of lines connected with dots, it is usually the case that not every dot is as important as the other. So we'll try to think about that at the later half. A big underlying message of this morning's lecture will be the kind of difference between a chemical equilibrium and a steady state. Biology operates on steady states. And if you can intuitively understand what these steady states are for different types of reactions, it becomes very useful. And finally, we'll just touch upon some of these things, probably later in the week. How, for example, you have some highly unfavorable reactions that still happen in a cell, or you have different ways to get to the same end. Okay. So first things first, of course, why metabolism? Really, it's the sum of all life-sustaining chemical reactions in organisms. So it's really the chemistry of life. And it seems irreducibly complex. You have a series of things connected with each other, but it's actually not. This is the complexity that can be nicely broken down into individual parts and points. And so I hope at the end of at least today's class, you start thinking that it's not one of those bizarre black box things. Okay. So why think about metabolism? This is again something Jonathan touched upon in his talk, I think early on. So we're looking at life and life processes, and of course, in the end, we're interested in form, function, tissues, organisms, but timescales for that are relatively slow. If you move further back at the level of cells, you have cells deciding to commit to different fates. These timescales, the determinants that decide cell fates are also slow, hours to days. We look at transcriptional networks, all of the transcriptional reaction. Even that happens in timescales of many minutes to hours. What this indicates is essentially the steady state that the cell has attained. Transcriptional signature or global transcriptional signature is a very good reflection of what that state the cell has attained and what it's going to commit to. Even before that, you have lots of things going on. Proteostasis, signaling. Proteostasis is basically controlling the amounts of proteins in cells even before transcription, signaling quick ways to carry out information transfer. And the thing that changes, that can change the fastest is metabolism, which is a primary function of the net chemical reactions in a cell. So the key point here is it's very dynamic, never static, and it's moving in some direction all the time. The moment it stops moving, you're dead. And if that's clear, hopefully we're all alive over here. And silence. We will increase some life entropy in a bit. Okay. Okay, so essentially what we're dealing with in cells is a constantly changing resource allocation problem. If you commit to making lots and lots of these kind of molecules, which we just covered recently, usually this becomes a signature for growth if you do all of the above. Making ATP, you're using ATP to make nucleotides, amino acids, lipids. You have parallel reducing power to carry out reductive biosynthesis. You're making proteins, you're making membranes. You're in a growth state. At the other end, you have all these possible outcomes: survival, autophagy, differentiation, quiescence. These are all terms biologists like to use. They're not that meaningful in terms of actually the chemistry of a cell, but it is very meaningful when you start thinking about what's happening over here. You're basically changing combinations of these. Either you don't have enough ATP or you have plenty of ATP, but the cell is unable to make enough lipids, or it doesn't have enough reducing power, A, B, and it's not A or B or C, it's A and B or C and B and C, so it's a complex network. But this is your resource allocation problem in the sense that at any given point of time, a cell or a tissue has to allocate different amounts of resources. But it's not something that is necessarily an active decision, right? Cells, for example, are not conscious, or if you reduce it even to just a cell lysate, there's no life in it in the sense of directed paths. What drives these are the fluxes through different chemical reactions. And the net flux through a reaction will determine how much of this is going on. And that depends on the kind of catalytic power you have inside, which is determined by how much of different types of proteins, or in some cases RNAs, are present to carry out the reaction. Okay. So there's kind of two parts. One is the bigger part, the meta part of how do you build up a cell with tens of thousands of molecules and small molecules, macromolecules. Yeah. But even before that, what really makes the cell are the combination of enzymes present, right? So do all of you know what enzymes are and how they function? So how do enzymes work? What's an enzyme? We'll leave the mic on in class and we'll just have somebody speedily running the mic on.
Is it
proteins?
audible?
Yeah.
Okay, so enzymes are basically proteins that catalyze mechanisms in order to either provide ATP or whichever is the final function that the cell is.
Why do we need enzymes?
Whichever reaction is to be, I mean, they are all intermediates, so they convert something into something and then.
Okay, so there are multiple terms here: enzyme, intermediate. Yeah?
Yeah. We have, there are some substrates on which.
Right?
And then with the help of the enzyme action, maybe there's a product formed.
Product. Okay. Hope you guys can see.
By themselves, maybe spontaneous. Probability of that.
Okay, so I can repeat what she says. She said reactions might be spontaneous, but
the probability of the reaction taking place might be very low because of the energy required.
Good. Okay, good. So essentially what you're doing You have entity A becoming entity B through some reaction, right? And any reaction by definition can go in both directions. Yeah. And there is a rate of a forward reaction and a rate of a reverse reaction, right? Good. What determines this? Yeah. You can just say it. If I hear it, I'll repeat the answer.
Well, the rate of the reaction can also depend on the concentration of the substrates
Yeah.
and the concentration of enzymes.
There is a concentration determinant of any enzymatic reaction. That's one important part of it. And then?
The free energy of the intermediate state.
Okay, good. We didn't put that in there. Free energy. What is the free energy?
So like when A turns into B, there'll be bonds broken, there'll be some associations it forms. So
Okay.
it's not a very stable state and it can't last very long, but it has to kind of go through that state.
Okay, so where is the enzyme in all of this?
I think like if you kind of make this edge a part of a cycle, you can an enzyme do this.
Let's say you put an E over here. That's an enzyme. Yeah, what does it help you do? Complete the sentence. You're there. I mean, it's there. It's there.
So it would basically take you through a series of steps where the free energy between each transition may not be that bad, that high. Or it could.
So, okay, we'll come there. Vaguely there. You want to answer that?
Say that enzyme reduces the energy of the transient or intermediate stage in some cases. So if it follows a lock and key mechanism.
Okay, good. Another thing came out. All these things that I want to cover in class have all come out. This is perfect. So I think I was thinking, okay, for this class, lock and key. Okay. Okay, great. So now we'll just keep all of this in mind and we will take a very quick tour through all of this. There's another one. Oh, there's one over there. Okay. Okay, so something, it holds. Okay, we'll see how much we can get there. Okay, so great. So enzymes are basically proteins, predominantly proteins. They can also be RNAs, which carry out or enable reactions to proceed efficiently in one direction or the other. That's the simplest way to think of an enzyme, right? So why do we study enzymes? This is a classic that Kornberg wrote in one of the last articles he wrote. You use it to solve. So I just like to have this in every class. Enzymes are important. People forget enzymes are important while doing all these other things in life. But you can solve large chemical and biological problems. It allows you to break down a complex problem to a specific component. It is universal. What you learn from an enzyme and from biochemistry is pervasive. And it actually allows you to say A does B. Genetics gets you to this point where it says, yeah, some gene is important for a phenotype. In order to say this gene becomes this protein, which does this reaction, and this reaction is therefore important, you need enzymology and biochemistry, and they are unique and powerful reagents. But of course, a lot of biochemistry had been reduced to studying one enzyme and one substrate at one time, which means the whole point was lost. Why was it lost? Because, of course, an enzyme has an inherent ability to function a certain way. If you provide enough substrate, it will make enough product. But it tells you nothing about the physiology of the system. So what we'll hope to take back from all of this is the importance of enzymes in a series of reactions in a pathway, because usually biology does not work from A to B and B to A. You essentially start somewhere And then you go to A, V, to C, to D. And of course, any of these B's can branch off to B dash and so on, right? And this can go here, and this can go here, and this can go here. And sometimes this can come back here, which means you form a network. And this is how biology functions. It's never operating at the level of one reaction, one product, and it's a continuum. So what determines the continuum will be some of these parameters, like this, and the presence of this. Okay? The direction of a reaction, right? So these are all chemical compounds. A will become B, B will become C, but C can also become B again, and B can also become A again in the backward direction. It is conversion, right? So what determines this is E1, E2, E3, and so on. So these are the proteins that determine which way it goes, right? Ah, yes, I did. E plus E. Thank you. Okay. Yeah, so the enzyme is always there. A very important point. It's actually not a minor point. Enzyme starts the reaction and ends the reaction as the enzyme. It's always there. Okay. Okay, so we'll just go over this one little bit of Michaelis-Menten kinetics, which is extremely useful and useless at the same time. How many of you guys know enough about Michaelian kinetics? A very small part of the class. Okay, good. So, this is Maud Menten, this is Michaelis. They were working about 100 years ago, more than that, 1910, 1915. And they essentially defined the properties of an enzyme. The key starting observation that they made was this. This is a classical Michaelian curve. You'll almost never see this in reality, but this is how this is how it should look if everything is Michaelian. Okay, what do you see here? What does this tell you? What do you interpret from this? There's a reaction rate and there's a substrate concentration, and there's this curve, and there's this Km, right? So for those of you who don't know Michaelis-Menten kinetics, what do you understand from this graph? I heard something at the back. You want to say? Aha. So after some concentration of the substrate, the reaction is almost saturated. Is it saturated here? Tends to saturate, but you can see this dot will extend forever, right? Endlessly. Okay, good. So first point is at some concentration of the substrate, the enzyme, the rate of the reaction saturates. So what does that mean? What do you mean by a saturated rate of reaction? That's absolutely right. What do you mean though? Now you have a cell, it's busily doing something, and you are saying the enzyme is operating at saturation. What does that mean? Rate does not increase anymore. That is the maximum you can do at that time. Okay? Rate is what? What is rate? Rate is the number of units formed per unit time. Correct? So how do you increase the number of units formed per unit time? This is the maximum the reaction can go. How do you increase the number of units formed per unit time? What can you do? Yeah, but the reactions are all determined by thermodynamics. There's a maximum limit to that. Now you are at the maximum. How do you increase the number of units formed per unit time? What can you change? There's no point in increasing the substrate beyond a point because you're at maximum. You can increase the enzyme concentration. Okay? What if you're operating here? What does this mean? Very nice. I like that word. Enzymes are not fully utilized at that time. What do you mean by you're not utilizing the enzyme? Enzyme's there. It's sitting. It's waiting. Okay, good. It's waiting for more substrate to come, which means it's operating at, this is half maximal. It could be operating anywhere on this curve. Okay. So this is really the essence of all the stuff that Michaelis and Menten put together through this reaction. Key observation, the rate of reaction is independent of the concentration of the substrate. It means the rate is a fixed entity. And this is observed only at higher concentrations of the substrate, which means when you do an experiment and you do this assay over here, this is what you'll see, right? Okay, great. And this is this, roughly, which derives to the Michaelian equation, which rarely ever works. dP by dt is the V max. This is the V max. Substrate concentration, Km plus S. Km is the substrate concentration at half maximal velocity, right? So now, this is very nice and abstract and mathematical, but what does it mean if you say your enzyme is operating roughly at its Km in the cell? What does it mean to you? You get the question? I'm saying now in this cell, it looks like the enzyme is approximately at its Km, operating at its Km. What does that mean? And so what does that tell you about the substrate? You know exactly how much substrate is approximately there at a steady state. So it actually tells you meaningful information of what is going on in a cell, okay? So we'll move a little ahead of this now, but essentially now you have many molecules of a substrate. Usually, in this Michaelian context, you have many more molecules of substrate than the enzyme. Yeah? As the enzyme becomes saturated at high substrate, its rate is at the maximum and it will remain constant. At low substrate, the enzyme will barely carry out a reaction. Could be due to various things. It doesn't bind the substrate or it doesn't release the substrate. We're not going to get into kinetics and all of that, but those are the reasons why it happens, right? So now, in a biological system, if you see lots of enzyme, does it have to be active? Because I'm always going to try to connect it back to an experiment you do or a cell that you see. You find there's a lot of some enzyme. Does it mean it's active? How will you know if it's active or not? Okay, to some extent. We don't need to worry about cofactors. So you see, where are cofactors in all of this? They aren't here yet. Those are very useful. We'll come to the next bit, which is allosteric.
You might have phosphorylation or something.
Yeah, phosphorylation is all allosteric. We'll come to that in just a bit. Exactly. You should see a change because it's dynamic. You should see a change in that per unit time. Of course, it's easier to think about this experiment than actually do this experiment. And we'll come to that maybe in some of the later talks. Okay. So some of these experiments are hard, but in reality, if you want to know if an enzyme or an enzyme in a pathway is really active, you need to know the rate of formation of its product. Okay, great. Okay, the other really important concept in there's a question back. I am defining active here right now as the formation of a product continuously happening. Okay. Of course, there are many ways to define activity, and that will be based on what you are looking to study. Okay, good. So how does an enzyme function specifically? See, so we just heard in the morning that there are hundreds, thousands, tens of thousands of molecules. Some of them are actually, they all look pretty similar. You know, glucose and fructose look pretty darn similar. How do enzymes discriminate between substrates? Of course, the answer is partly here, but in reality, so this is again an abstraction, but in the end, you saw how crowded that cell was. It's packed. It's like literally packed, which means all the enzymes are present and all the molecules are all present at the same place at the same time. So what's going on in there? How do you get a discrimination. Yeah, so we have affinity. What does that mean? Affinity is not a property of activity. Affinity is how well A binds to B. Yeah? And what does that mean? So what determines more probability that they will come together? Sometimes most of the time it's non-covalent. Keep this in mind. We will hopefully discuss this as we go along. And as we get into some practical components, we'll see how this works, okay? So anyway, the original idea was that this enzyme was this perfect, this is the original Fisher's lock and key idea, that the enzyme was this perfect shape and the molecule was the perfect match to this perfect shape. And so only one molecule could fit in one cavity, like a lock and a key. And so you would form an enzyme substrate, and a product. Of course, this turned out to be quite wrong. Essentially, what you have is what we still call an induced fit. This was what Koshland proposed many years ago. Essentially, what you have is, let's say you have a molecule like this. The protein doesn't exactly look like that in its cavity, but it has some space where this can bind. And as it binds, the protein changes its conformation to fit quite correctly. So that's the induced fit. And then if it's an enzyme, it'll carry out a reaction. The reaction will now, once the reaction is complete. So for example, if the reaction is to break this molecule into these two molecules, the moment it is broken, now the cavity no longer fits because now you have two different molecules. And so they will be released. You get it? And essentially that's how a lot of enzymes operate or function. Okay, good. So that's just at the level of basic allosteric, means to identify and bind molecules and so on. But then, as we understood regulation, the next really big jump that helped us make sense of a lot of biology is allostery, which actually helps you understand how you could carry out so many different reactions together in different ways at the same time and talk between pathways or between reactions. What is allostery? This is absolutely central to our understanding of regulation in biology and in metabolism, starting, but everywhere. So what is allostery? Yeah. So if it's an enzyme, it is regulation of an enzyme at a site that is different from the active site. So if you just see here, what you have here is what is an active site. It is the place where your substrate can go and bind and carry out a reaction. An allosteric regulator is something that won't bind here and is not part of the reaction at all. It is in fact unrelated to the reaction completely, but it will bind somewhere here or here or here. And by doing this, it allows these induced fits or conformations to change to make this entity active. Okay? And we'll now, in a little bit, we'll discuss how that builds in regulation into a system. So I'm just putting this here because I think, if you're a biologist, at some point of time, you should go back and read these papers. It's actually a whole issue of JMB. This is the key paper, but there are, I think, 8 papers Mono, Shango, and Jacob had in this one issue of JMB, which explains allostery. And it's beautiful. Essentially, what it says is that certain proteins at critical metabolic steps are endowed with functions of regulation through the agencies of these proteins. And a given biochemical reaction is eventually controlled by a metabolite acting as a signal rather than chemically essential for the reaction itself. Okay. Now, this idea repeats itself continuously in different templates in biology all the time, everywhere, at the level of proteins and molecules. And these are really the defining features of allostery. Now, so you have different parts of a protein. One is the site that is involved in the reaction. The other one is unrelated to the reaction. But so the active site binds the molecules for the reaction. The other site, which is the allosteric site, binds something else. And that something else can control the reaction, whether that enzyme is activated or not or whatever. Now, today we look at a lot of signaling in terms of modifications, post-translational modifications, phosphorylation, ubiquitination, whatever else, right? All of those are essentially allosteric modifications. What they do is they bind at a different site from the catalytic site to enable that protein to acquire certain function. This is actually just taken from the paper, but I've just put the citation here. In your free time, it's actually a pleasure to read. It's a very long paper, it's very mathematical, and it's beautiful. So, allostery, is it for activation or inhibition? Both. Because conceptually, it's agnostic to activation or inhibition. Oftentimes, students think allostery is only for activation, but it's not. It works just as well for activation or for inhibition. How does allostery work? There are essentially 3 different modes of allostery that are universal in every protein studied. One is the active site opening. So, for example, this is an allosteric site. Some molecule entity has to bind there, and only when it binds there, the protein undergoes a conformational change, allowing this, in this case, the substrate to bind and carry out a reaction. How will this work for inhibition now? Yeah, closing this, right? So you can see the exact opposite, where the allosteric inhibitor binds and it prevents the substrate from binding. Okay. The other very common mode of allosteric regulation is to form the active site itself. So here there is an active site, but it's closed. The other here, there is no active site unless the allosteric regulator binds. In which case, then you get an active site that is perfect for this molecule. Okay? How will inhibition work here? Same way, but opposite, right? What it will do is when this binds now, you will create something like this. It means once you bind, you no longer have an active site where the substrate can be. And then the third way this can happen is how? You have to guess now. Exactly. Okay. So this is the other way. What you can have is a molecule that will bring together either 2 subunits or 2 entirely different proteins to make them a functional enzyme. You can give it a fancy name. You can call it a molecular glue or something. But essentially, it's bringing 2 proteins together to carry out a reaction. And this can work, of course, both ways, because it could also prevent the binding of a protein with the allosteric regulator. So now we think about this. So we think about this. How is this now useful in a biological network, biochemical network? What does this allow you to do? So I'm just going to, this is allostery. At this level, this is all you need to appreciate. But how does this now leave you with useful biological insight? Or does it leave you with useful biological insight? Way back then. Yeah, so if we come back here to A to B to C to D or something like that, you will have allosteric regulation potentially at any level. It can happen to any part of the reaction at the enzyme level, right? If you have a physiological molecule, some molecule that's present, that is usually not there, but is, let's say, made in some reaction over here, right? Supposing you're having a reaction, it's going on too fast. The cell needs to turn it down. What will it do? It will inhibit. One way to inhibit is with the product. But if you are biasing towards creating reactions that are favored in one direction, this is inefficient. What would be more useful is to make some molecule somewhere later, which can go back, let's say, allosterically in one of these modes, bind this, stop this from functioning, you'll reduce the rate of the reaction. Right? So that's a feedback loop. You could have the opposite. You need to have more of this reaction because there's too little of it in the cell. The cell needs more. What would you do? You could take something maybe later on, or maybe something early on, which can go back. Usually it'll be something early on if you want to activate a pathway. This will go and say, activate this enzyme. By doing that, this becomes more active. You start getting more of this and then more of this and more of this in order to make more product for whatever the cell is trying to do, trying to make more lipids, right? This allows you a feed-forward reaction, an amplifying reaction. So with allostery, so to summarize, there are three things going on at the same time. One is that enzymes have some affinity to its substrate. That is determined in part by this stuff, right? In addition, so that is inherently in itself. In addition, it's regulated at different levels with various modes of allostery, where you use a physiological signal to control either an increase or a decrease in the reaction. Yeah? Question? Mhm. Yes. To control the rate of the reaction. That's correct. That's correct. So what are the time scales we talked about? Metabolic reactions are, I mean, faster than seconds to seconds to minutes. That's the time scale. The whole process of transcription, translation, stable protein is minutes to hours, which means if you need rapid so for physiology, this is important because some reactions only need slow regulation. If you don't worry, if it's not some reaction that's operating at a very, very high rate, you need only small amounts of it to be regulated, you're perfectly fine just sticking to protein synthesis or degradation, whichever, right? If you're operating at fast scale reactions that have to be done continuously, ATP synthesis, for example, I mean, you literally can't afford to stop making ATP, right? Most of those would rather be regulated in some combination of this, typically. And of course, at longer time scales, at the organismal time scale, it'll have a combination of both reaction regulation as well as protein regulation. As the cell differentiates and becomes a different cell, it'll express different amounts of that protein. It doesn't need that much of it, so permanently it'll express a lot less of it. And so the rate of the reaction will remain low. You have to use a combination of both. Make sense? Okay, great. This one. This is the paper. Wait. Yeah, that's the citation. But this is the whole issue of JMB. The entire issue 12 of the 1965 JMB was Jacob and Monod's complete description of every aspect of allostery. This is the main paper. The others were not only on allostery. I mean, they used to do that in those days. The whole issue of a journal. Two authors. Insane. Okay. So now we come to what reactions look like in reality. So I showed you a perfect Michaelian curve, right? And you can see that for some enzymes. But now the Michaelian curve we drew was something like this. Right? This assumes perfectly linear relationship between the enzyme and the substrate till you get here. If you have an allosteric regulator, what will this curve look like for the enzyme, for the enzyme substrate, for the reaction? Will it look like this? Right. So now let's say we have an allosteric activator. What will this curve look like? Yeah. You might have something like this. Right? So now what has changed? Obviously the Km is shifted this way. But also experimentally, when you measure it, you think you've changed the Vmax because you're getting to this point very quickly. In a Michaelian enzyme, you'll get to Vmax very late. But here you've almost reached something close to Vmax. So it actually doesn't change the Vmax, it's still the same. But in an experimental assay, when you set up assays as an experimentalist, you have to watch out for these things. You suddenly see this big spike and say, oh, this is insane. We've increased the Vmax. You haven't. You've just shifted the Km to the left. Yeah? How will an inhibitor look like, an allosteric inhibitor? The activator is easy usually. How does the inhibitor look? In the same reaction, you've added an inhibitor. What will it look like? Well, not to this, but let's just say we're starting with an allosteric inhibitor. What will the curve look like? It will shift to the right. What will it look like? Of course, it will shift to the right. Good. So, going on here. And that's right. So, presumably, it will be reversible. Yeah. You'll end up start to see things like this. Yeah. In theory, this is not straight. It's still going towards Vmax, but this is an infinite Vmax. Yeah. So that's how you have a lot of regular. Yeah. So now it's no longer Michaelian. When you have an activator, you no longer follow Michaelian. You follow this. It's a very sharp sigmoidal. And of course, we're not going to get into cooperativity and all of those things. It's well beyond scope of this class. But essentially, what you'll have is a very sharp sigmoidal, and you have ways to calculate cooperativity. But you can see how powerful regulation can happen in this. Okay. Now, I'm not going to get into free energy too much. It's just a kind of an important concept to think of. Again, not necessarily like a physicist, but more like what does free energy mean for biology? So all of these essentially, remember, is that your direction of a reaction is determined by the Gibbs free energy. Okay? And so the parameters here are, you have entropy. What is entropy? What is S? What is this entropy thing? Randomness in the system. You stop being random, you're dead. So it's random. That is the randomness in the system. What is the free energy, the G? So what is the free energy of a system in just its simplest definition? The amount of energy you could imagine is available to do work. Yeah. There's enthalpy. What is enthalpy? Yeah. Yeah. And then you have the delta free energy, delta G. That is what? Whether net energy is dissipated or absorbed in a reaction, right? So essentially what we really care about is the delta G in a reaction. Why? Of course, I'm not going to derive this. You can see the textbook. It's relatively straightforward. But you'll get a delta G is delta H minus T delta S. T is, of course, temperature. What does this mean? Practically, what does this mean? So what does it mean to have a very high delta G? Yeah, so if you have a very high delta G, is it spontaneous or not spontaneous? I is positive, right? Yeah, so you have a delta G of very high. Non-spontaneous. So what does that mean? Non-spontaneous is you think it won't happen. As in, if you just have A, you're interested in going to B. If the delta G is very high, very, very, very little of this is going to happen, right? Now, is it possible to have a high delta G reaction in biology in a cell? Good, wait, we'll hold off on that second part. So, first part is yes, there are many reactions in a biological cell which are ridiculously high delta G, which means if you just take A, it will never become B, unless you're raising the temperature to 1000 Kel, something ridiculous, 1000 centigrade, which is you combusted your substrate or whatever. So how do you have a reaction that's high delta G in a biological system? Yeah, yeah. So what she said is you have an enzyme that will enable the reaction to go forward, but can it change the delta G? Yeah. Let's say this is delta G is G. Okay. So what the enzyme does is reduce this so that this can happen. But still, if you look at this is time. Um, this reaction still requires a negative delta G. So inherently alone, no matter what enzymes you put, you still can't have a positive delta G. But biology is full of high delta G reactions. Okay, ridiculously high delta G reactions. So how can we have it? You said something here. Yeah. So the only way you will have it is if that reaction is coupled to another reaction which is ridiculously low delta G, so that the sum of the two is a negative delta G. Yeah? Which means you have multiple reactions that alone are impossible. But all of those, whenever you find one of those, you need to find another coupled or a set of coupled reactions, which are all highly favorable reactions to give you this reaction. So now how this might work is this. Let's say this is very hard. Okay. This is like plus, delta G, like highly positive. Metabolism is not static. Supposing this is here, that's highly negative delta G. Then you have another one, highly negative delta G. These reactions will literally happen the instant a little bit of this is formed and will give you enough free energy to lower the total free energy to a negative, which means this will start moving forward. Okay? You have multiple examples of this happening in a metabolic network. This is why delta G is useful. We don't do anything more with it. We don't worry about the absolute numbers. We don't have to calculate it most of the time. But it's only useful when you put it together as a part of a whole network. Okay? Okay, good. Yeah. So we'll just complete this thread. How do you know if a reaction is spontaneous or not? Essentially, what you have in a reaction is this, right? Some concentration of A and some concentration of B gives you something of C and D. This is a standard reaction. What is the equilibrium constant over here? This you should know. I mean, if you have a reaction, simple reaction like this, what is by definition the equilibrium constant? This by this, right? Yeah? So you'll get that, essentially this. When you look at this, if you see this experimentally or you observe something, you don't know the equilibrium constant, but you start seeing different amounts of this and this, you can actually guess what the equilibrium constant would be like. But what does a large K equilibrium mean? If you have lots of this and very little of this, the reaction is tending towards being spontaneous. Yeah? Correct? Okay. So, now what is the main energy currency in a cell? ATP. We all agree to that, right? Okay. How do you make ATP? Putting a phosphate group constantly. Yeah? What's the delta G of this reaction? Is it positive or negative? We could actually work this out if you had a long class, but if you look at it, what you're doing, you're adding inorganic phosphate. Is it minus or plus? It's positive. Is it slightly positive or very positive? Actually, this number is a pretty standard number, right? What is it? Very positive, right? So this is actually the real equilibrium constant. This. Okay. So if you had just a test tube system, you should do this now. Take a minute. How many molecules of ADP, how many molecules of ADP do you need to have to go in the opposite direction here? How many molecules of ADP and PI? Let's do this. I mean, this is all units of 10. Give me a number. Forget the 2. You just keep 10 to the 5th. Will I have a sip of water? So I mean, this arrow is in the other direction now. If you want to go from here to here, the equilibrium constant is this for this reaction. Give me a number. Give me a number. Number is important because the next thing I'm going to tell you is going to be dramatically opposite to this number on what's happening in a cell. Right now, we're starting with only this reaction and nothing else. Okay, we can even take this reaction. It's the same idea here. This would be very positive. Adding of inorganic phosphate to a molecule is a ridiculously plus delta G reaction. Okay. But of course, we're looking at only this last reaction, ADP to ATP or ATP to ADP, right? So this is highly favored in this direction. But okay, just look at this. If you're struggling with the math, if this is the reaction, should we have any ATP in the cell at all? These are hard numbers, right? You can't mess with these numbers. But according to this, you practically can never have ATP in a cell. Do we have ATP in a cell? We do. How much ATP do we have? Is it more or less than ADP? It is at least 10 times higher than ADP. Okay. So in every known living system, doesn't matter if it's an E. coli or a fancy neuron, the approximate ratio of ATP to ADP is 10:1. So there's 10 times more ATP than ADP, right? And we consider a cell energy starved if that ratio changes to 9:1. That's the window we have, barely. That's a ridiculously small window. But I have just said that this is impossible. So how do we have 10 times more ATP in a cell than ADP? That is a kind of a way, but how do you get so much? What needs to be happening at any given point of time? That's part of it, but that's not it. Presence of enzymes is useless here because enzymes, it's just so skewed the wrong way. Two things have to happen. One is your rate of consumption has to be so high as to keep this forward reaction going, right? Like where was this? Yeah, here. Something far away is taking away so much ATP that this reaction is moving forward. And the second is you have a series of negative delta G reactions coupled to this to give you the net gain for ATP, which is what gets you to how energetics happen. Okay, so that's, somebody said proton motive force. We won't have time to do that, but yeah, but that is part of this answer. Okay, good. This one, yeah. So the same idea then. So what do you think of first when you think of generating ATP? Why does it? No, it'll continue. You mean here, ATP, triphosphate. I mean, we've already got enough of what we need to do with the triphosphate. Oh. I mean, just the delta G's keep increasing and increasing. In theory, you could have a tetraphosphate. No. So you have, for example, polyphosphates. We have plenty of polyphosphates. They're very abundant molecules in the cell. They can also, in some contexts, work as an energy source. But biology is happy with ATP in this cycle, going from ATP back to ADP and so on. Because this delta G breakdown is perfect for all your coupled reactions. You're getting, each time you have a coupled reaction, you're using this energy of the breakdown to carry out some other reaction. So there is no like, it's perfectly fine. You have polyphosphates, plenty of polyphosphates. Yeah. Then? No, it's additive. This is a plus delta G, approximately the same and approximately the same. That is the delta G of putting one free phosphate onto an existing molecule using inorganic phosphate. Okay. So the key here is you have multiple coupled reactions with this reaction, and the net delta G has to be sufficiently negative to drive this forward. And part of the answer is also the rate of consumption. If you keep consuming more, you have to keep making more. So these two things are coupled together. Okay. So the key, way back. Forward and the reverse reaction. Is that the question?
Yeah. No, I'm thinking about AMP to ATP and ADP to ATP. If the delta G's are similar in both the cases, why do we stop at ATP and not go to AMP? I mean, that should be the last stop then, no?
I mean, the why is like a philosophical question. I don't know. This is what we have. But I mean, if you go from ADP to AMP, also you'll have a similar energy release.
So in a test tube kind of a situation, the final state that I'll see.
In a test tube, we'll never make ATP. If you just keep AMP or ADP, you will never actually be able to make ATP in a test tube without everything else, without all the coupled reactions going on at the same time.
No, if we start with a pure solution of ATP, let's say.
Yeah.
Then
it breaks down. So if you start with a pure solution of ATP, it will nicely be hydrolyzed to ADP, and ADP will get hydrolyzed to AMP. You can see that very nicely. In fact, people have done this experiment way back 50s or something like that. Nicely hydrolyzes back. And then it starts accumulating as AMP. The AMP, this is actually reasonably stable. It stays there. It doesn't go that quickly back to tennessine. Yeah. Okay, so most of energetics, when we think about it, it's breakdown of glucose. Yeah? You agree, right? Most of you, first thing you think of. This one person is thinking of, no, I'm only on a pure high-fat diet. I mean, we'll discuss that later. Okay, so this is the first reaction of glycolysis. Even before glycolysis starts, what do cells do when they take in glucose? They put a phosphate on it. Why do they need to do this? The reasons are purely biological. Glucose is an uncharged molecule. If it remains uncharged, it will leave the cell. That's because of the little lipid bilayer that we just heard in the previous lecture, right? You want to keep something inside the cell, you put a charge on. So they're putting a charge on it, and you put it with a phosphate. If you just, this is the pure chemical reaction, glucose plus Pli is glucose 6-phosphate. This is a highly positive delta G. But now with all that you've learned about delta G, how is this possible? Because essentially the instant glucose is taken up, cells put a phosphate on it. So what must they be doing? Part of it is that, but to consume it, you have to first put the phosphate on it. So there's a chicken and egg, but to put the phosphate on it, this is a highly positive delta G reaction. How would they do it? They'll couple it to this reaction. This reaction is a highly negative delta G, right? So it comes back to why we are able to have this constant cycle of ATP production and utilization. This is your delta G. Reaction 1 is a 13.8, highly positive. But this breakdown, this is the famous number that you should probably remember. The delta G of ATP hydrolysis is around 30 kilojoules per mole. So now you get a nice net negative delta G. This reaction goes forward, right? But we have to do it. This is a useless reaction for the cell in terms of energy. It's wasting energy, but it's done it. Why? Because it needs the glucose to get more energy, right? Yeah. Okay. So what we're going to do in the last 15 minutes or so, yeah, 20 minutes, yeah. What we'll try to do now at the end of this class is to try to eventually derive glucose breakdown from first principles so that you don't have to remember those horrible reactions of the pathway, right? The few biologists over here must have struggled through their biochemistry 101 memorizing glucose 6-phosphate, fructose 6-phosphate, 2,6-bisphosphate, all of that. You don't need to know that. And we'll see why. It all comes down to the eventual net need to get some ATP at the end of all of this work. Why do you get this ATP? To do useless things like utilize it to break down itself. Okay, so that's what you get ATP for. So now biology, and I hope you've already appreciated it, all these enzymes are operating in a series of pathways, which means biology operates at different steady states. So what's a steady state versus an equilibrium? Who can define this? What's the difference between a steady state and an equilibrium?
So an equilibrium state is a special case of a steady state. In both cases, there are no macroscopic changes in the system.
An equilibrium is a special case of a steady state, but what is an equilibrium?
So the steadies, there are, to maintain the steady state, you don't have to put in any energy. It just is the way it is.
Okay, little abstract. Anyone want to simplify this? Okay, so she's saying, in case you couldn't hear it, steady state could be given, could be coming from a lot of different substrates and products, while an equilibrium is what? Yeah. Equilibrium in chemical terms is rate of forward reaction is exactly equal to. Okay. What is a steady state? There's one back there. Yeah.
Steady state we can say as a dynamical system, but equilibrium it states that like we don't put input energy or output energy, it's like that. But in steady state we can we have to constantly input energy to maintain that state also.
Okay. Okay. So imagine a bucket. Okay. Let's say water is going in. Initially, when water goes in, what happens? And water can go out over here. Okay. Initially, when water goes in, is the rate of, these are exactly the same size holes and everything else. When you start filling the bucket, does the water go out at the same rate as the water coming in? Oh, this is not a physics question. This is like open the bucket at home. Okay. It doesn't, right? What happens first? Let's say these two are exactly the same. What happens first? It goes out slowly, which means what's happening inside here? It starts to fill, right? And at some point, after it fills up to a certain level, which can be calculated, at that point, this and this will be equal. That's why it stops filling anymore, correct? But it does fill up to some point, right? This is a steady state. This is a steady state, which is what is happening in a biochemical network, because your input could have a long series of reactions over here, going into A giving B, which is continuing to give something else, right? Your steady state is when this input and output, you are measuring this and this. These two are the same. But what's happening here need not be the same, right? Rate of forward reaction is not the rate of the reaction, reverse reaction. That's your steady state. So all biology is operating at steady state. So now if you think of ATP production and utilization, you're operating at a certain steady state. Other reactions are operating at a very different steady state, which means there are reactions that are operating, when you think in terms of numbers, you might be operating at, pico, nano, micro, milli, mole levels per unit time. You can have different steady states at all these levels. Different types of reactions only need to operate at these different levels. So now there was an earlier question on, for example, using purely transcriptional translational regulation. Those are all very effective for reactions that are operating at relatively low concentration levels. You don't need, and because the rate of forward reaction is slow, the rate of reverse reaction is slow, the rate of input and output is slow, everything is relatively slow. You're operating at those time scales, you could rely completely without having an allosteric enzyme, without any of that. You're operating at these very large rates, thousands, tens of thousands of molecules per second. See, one of the things I like to do is to calculate how many molecules of glucose a cell uses per second. If we have time, we'll do it. Ridiculous. I've just consumed so much glucose right now, speaking all of these things, that these numbers are staggering. But that's the point. So you can see you have very, very different steady states. And high consumption reactions tend to operate at usually at a higher steady state with really high rates of this and this. Yeah, but you measure only what you measure. So we come to other things that the cell enables. How do we have these highly improbable gradients, like with glucose 6-phosphate, right? You have a membrane, you take it in, it can't go out. So you can maintain reasonably high concentrations of glucose phosphate. Second, when you're carrying out things like transport from lower concentration to higher concentration, what do you need? You need to create effective negative delta Gs to carry it out. If you have a bunch of transporters, you'll do it in 2 or 3 different ways. What will you do? You'll use a lot of ATP, for example. Or you will couple that to something that is high concentration inside to have an antiporter. You'll have something coupled to another something, take one in, take one out, which you see all the time in biology, right? Coupled channels, coupled transporters, and so on. So that's how you achieve steady states in biology, many of which would be impossible without the creation of these things. One is barriers in systems, the other one is active energy consuming processes. Okay. So last 10 minutes, we'll just get to, we'll just do glycolysis. There's a lot of pathways in energy metabolism. Primarily the oxidation of glucose, and this is the oxidation of glucose, right? Glucose plus oxygen gives you 6CO2 plus 6H2O. That's just the chemical reaction. What else comes out of this reaction? If you do all of this, how much ATP do you get? Complete if you have net 2 molecules, then you won't be at CO2. You'll be at pyruvate. Around 36, right? How do you get to this number? Right? Typically, for simplicity, we'll go only halfway, from 6 carbons to 3 carbons. which is glucose to pyruvate. Pyruvate, or you could go all the way to lactate, but no difference in the energetics. How many molecules do you get? You know, you just said it, 2 molecules, right? And how do we get to this number of 2? And is 2 molecules a lot or very little? So most cells, you have 2 ways to make energy, what she was alluding to. One is the glycolysis part. The next part is entry into the respiratory cycle, the tricarboxylic acid cycle. Together, if you oxidize glucose completely, you will get carbon dioxide plus water. Okay? You do all of that, you will get about 36 molecules of ATP. If you do it only halfway, up to pyruvate, you'll get only 2 molecules of ATP. Is that a lot or very little? Not a lot. It's not a trick question. But you'll find that in a lot of systems, a vast majority of cells rely primarily on glycolysis for their energy. How do they do that? They have to do a lot of glycolysis, right? Which now gets you to think about how much glucose is being consumed per cell per unit time. It has to be a lot. Let's just derive glycolysis. I like to do this, so we'll do this. So what do we start with? This is glucose. What did I say the cells have to do first to keep the glucose inside? You have to add a phosphate, right? And that is just a requirement to keep glucose in the cell. And to go from here to here, you already now know this. Is this going to take energy or release energy? It's going to take energy. Good. This is the reaction. Is it a positive delta G or a negative delta G? The reaction. And the basic reaction, the chemical reaction is highly positive, but the way you make it negative is by utilizing ATP. So now it's very clear. If you have to see this ridiculous reaction anywhere, you see a molecule like this and you put a phosphate on it, very hard to do. Most likely way you've done it is by consuming ATP. You've done it. Good. So now you've made it negative, you've moved it forward. Now we'll also come back to this beginning idea of regulation when we're thinking of pathways and the need to amp it up or amp it down. Do you think this step needs to be highly regulated? This enzyme is the enzyme, hexokinase, the famous enzyme. Does it need to be regulated? Yes or no? Opinion? That's right. You want to phosphorylate it all the time. So good. So now when we're looking at order of reaction, we didn't really get into it, but many of you should know, you're physicists. You know what a zero order reaction is, a first order reaction is, and a second order reaction is, right? What is a zero order reaction? That's correct. Which means this will be units per time. It will look like this, right? Straight line. With a concentration. Of a concentration of a product we can put. It will be like this. It will be like this, sorry. Concentration of product is going to be flat over here, right? Why? Because it's operating at close to its Vmax, right? And that's what happens in reality. Hexokinase is operating at its Vmax, which means the amount of glucose and the glycolysis rate that is going to be determined in the cell depends on how much hexokinase is there. So now if you have to guess which tissues have the highest amount of hexokinase in the body? Liver primarily, that's your primary site of glycolysis, maximum. Next is muscle. They have the highest amount of hexokinase, and the next is brain. For reasons unknown, brains use a vast majority of the ATP. But it's easy, no other regulation is needed. It's a zero order reaction, right? After that, the next step. Now this everybody knows and memorizes if they do glycolysis. You go from glucose 6-phosphate to fructose 6-phosphate. Why do you do that? Why do you need to do that? The chemical formula is almost the same. But what changes? If you draw it this way, it becomes very clear. What is this? What is this? Hmm? You can think about this over lunch or whatever when we get some more glucose, but you can just see this little shape change, okay? Why would you want to do this? And before that, what is the reaction here? Any change in the phosphates over here? Not really. You're just moving it around, right? So is this going to be a high delta G or a low delta G? Hmm? There's also something in between, it's like a net zero. This is going to be an approximately net zero delta G. Not very hard to do, it'll happen. If you have an enzyme, it'll just happen. Okay, so it's done. What was the purpose of doing this now? How do you break down molecules in chemistry? You try to create a symmetric molecule so that it can break in two. So all you've done from here to here is just change its structure to make it something like this, which can become perfectly symmetric molecule by putting on another phosphate. The moment you make something like this, it's unstable. Okay? That's a longer answer. We can talk about that over lunch. But I will explain this to you, okay? Because we haven't gotten into chemistry at all in this class. Okay. But you basically now you want to put on a phosphate. Is it hard to do or easy to do? We talked about it in the beginning. Always putting on a phosphate is very hard. That means what you have to do over here, delta G's of the reaction itself is going to be high or low. Reaction delta G would be very high. The only way to make it really low is by using ATP. That's exactly what you do. Okay. Now that you've done this, which means what have you done? You've wasted 2 molecules of ATP over here. You've used up a lot of ATP to get to this point. Cells are doing this all the time. But what have you done here now? Once you've reached here, you're in great shape. Because the next thing that's going to happen is what? Molecule like this, you can see it's colored nicely in blue and red. It's going to break into two not exact halves, but two very similar halves. You get these two molecules. This one is going to have a high or a low delta G. Yeah, it's a slightly low delta G. This will almost happen spontaneously. Okay, this is very easy to do. Now the cell is in business. After all this work, it can break down this fructose 1,6 bisphosphate. From here, what do you do next? Those of you who've studied a little energetics, now that, see, all this time you've not made a single molecule of ATP. You wasted energy to do it. Now we have to find a way to make ATP. What is the advantage of getting these 3 carbon molecules? So you've gone from 6 to 3. One molecule has become 2 molecules, one of which is actually useful. This is the one that's useful, G3P. Why is that? See the difference here. Yeah, this one is always more reactive and easy to work with. What did I do here? This is an aldehyde, glyceraldehyde. Glyceraldehyde 3-phosphate. This can become a bisphosphate because now this can react with inorganic phosphate. This is a highly favorable reaction. Finally, now we're coming to use something that is abundant and easily available, which is inorganic phosphate. And this is a net negative delta G reaction. This will happen very nicely. And this is actually very famous enzyme for different reasons. It's GAPTH. Most of you use it as a loading control. Those of you who do biology, it's actually an important protein. Okay, it's not just to measure how much is there, but this is present in huge amounts. This enzyme, also because phosphate is present in huge amounts. As long as you're steadily supplying this reaction will keep happening. And now finally, you're using free phosphate without burning ATP. Okay. Now that you've got a molecule like this, what have you done again? You've created kind of symmetry, put phosphates on two different ends. Yeah? So you've used inorganic phosphate. You'll now convert this to 3-phosphoglycerate. And when you do this, what have you done here? Basically, you have a symmetric molecule, 2 sides of a phosphate. And because this reaction is nice, And this now is ready to leave. Once you put the phosphate on, it's ready to leave. You can actually put the phosphate onto ATP. This reaction from this molecule to this molecule is favored. What does it tell you about this molecule? This one is at a higher energy state than ATP itself. That's why it's able to transfer the phosphate on. Now you finally got your ATP. And if you've just done it this far, your balance sheet is zero. You've used 2 molecules here, but you've made 2 molecules of these to give you 2 molecules of ATP. You're at net zero. So now you've just broken even, and now you need the payoff on your investment. Now you just have to keep doing the same thing. Try to start creating some kind of symmetry or make a molecule that will leave. In this case, if you have a hydrolysis reaction, you make phosphoenol pyruvate. Now this is another molecule now sitting with this phosphate sitting there. This phosphate cannot just sit there. It has to leave. Every time it has to leave, you put a phosphate onto ATP, which means this delta G is again highly, highly negative. This is at a higher energy state than ATP itself. So it can transfer the phosphate to ADP and give you pyruvate. So now what you're getting now is payoff ATP. All the ATP that are being made here, 2 molecules per molecule of glucose, is giving you a net gain of ATP. Cool? So essentially, this is glycolysis. You don't really need to memorize all of these things. What you're doing is consistently the following. First, you're trying to create a molecule that can become a perfectly symmetric molecule like this that will break. Once this breaks, you're creating again smaller molecules where you can put on phosphates that will then donate that phosphate to ATP over here, ADP over here. Back to that question.
So I'm sorry, my chemistry is really bad. Is it supposed to be obvious from the structure of phosphoenol pyruvate that the phosphate group is not stable there?
This one, yeah. This is ready to leave. It's going to fall off.
Is it supposed to be obvious from the structure? If I see
From
the
this,
structure?
yeah. If you look at something like this, it will leave. But we can talk about a little bit of the chemistry. But yeah, this is very, very straightforward, simple chemistry. You see a molecule like this phosphate, you put it in water, you put PEP in water, the phosphate will leave at some point of time. Yeah, it's ready to leave. Okay, so that's glycolysis and net gain of ATP. So now this is what the cell is doing continuously all the time. So now we come to regulation, all that stuff that we talked about earlier. If you didn't know anything about how fast is the reaction? And nanoseconds, milliseconds in a cell. So the fastest observed, so like in a cell, the cell with the highest rate of glycolysis is yeast cell, Saccharomyces cerevisiae. If you and so your substrate is at tens of millimolar glucose, that's what you're doing. You try to do like a label saturation experiment. The whole reaction, so you have 10 millimolar glucose, you spike in 10 millimolar labeled glucose, that reaction saturates in about 2 or 3 seconds. The whole reaction, 100% saturation, something like that, or 50-50 saturation, which means reach steady state. Sorry. Everything, all put together in the cell. So this reaction is really fast, milliseconds per unit glucose. So you're consuming, a single cell is consuming tens of thousands of molecules of glucose per second, actually. That's how fast it's going, super fast. Of course, cancer cells are roughly in that ballpark. E. coli is very much in that ballpark, very fast. There are some cells which use it very, very slowly, and those are, small subset of cells. I mean, if you just do the numbers, just the cytoskeleton and the ribosome is consuming so many molecules, that many molecules of ATP per second in a cell. The whole cytoskeletal motors and ribosomes together, just those two. Forget all the other reactions in a cell. Yeah. Okay, so which one of these, if you predict, will be regulated? If you have to regulate the whole of glycolysis, which ones would you want to control? And in which direction? Like, do you want to activate it or inhibit it, or both? Because usually physiology is beautiful this way. It doesn't mess around with useless stuff. So if you have to guess your two most regulated enzymes or one most regulated enzyme, what would you pick? You are an intelligent designer. We'll end with this. This one. Yeah? Yeah? Okay. That's a. Why did you pick that? Yeah? So this step you would guess is the most regulated, right up front on top. If you have to guess, and it is. The enzyme that does this, phosphofructokinase, is the single most regulated, allosterically regulated enzyme known so far. Okay. So now how would you want to regulate it? What would you need to do if you want to increase the total flux through glycolysis? What would you do to this enzyme? Oh yeah, sure. Right here, yeah. No, just one. One phosphate, one phosphate. Oh no. So the reaction is favorable as long as the net delta G is negative, but you're not perfect. You're wasting a lot of energy. No, there's only one phosphate. No, law of conservation of mass is also there. It can't couple to the inorganic phosphate addition because that delta G is too much. It won't come. Yeah, waste. Huh. So this is the most regulated step. How would you regulate it now if you want to increase the rate of glycolysis? What would you do if you were a cell? You were an intelligent designer. You were whatever you were. One simple thing is increase the amount of this enzyme, which is kind of futile because this is already operating at its Vmax and zero order kinetics, no? So what else would you do? You would create an allosteric activator. But what allosteric activator would make sense? What is the cell trying to do in the end? Make enough ATP, right? So a very basic allosteric regulator could be something like ADP. You're slightly starved of energy. So ADP can activate this enzyme by a little bit. What would be even better? Because I told you the ATP-ADP ratio doesn't change that much. It means it won't be a great allosteric activator. You could also have pyruvate itself controlling it. So if you have small amounts of pyruvate being made, if this binds that enzyme, it could activate it, and that happens. So it is a modest allosteric activator of PFK. If you want to do even more, what would you do? So AMP is done. It is the worst allosteric activator. It is an allosteric activator, but it's a weak one. Pyruvate is a better allosteric activator. It can activate it. What else can activate it? If you really want to ramp up the rate of production, AMP, yes. Pyruvate, yes. Little.
How about
ATP, what do you think ATP will do to this? Should it inhibit it? No, if you have too much ATP, you should inhibit it. ADP activates. So ADP, yes. ATP mildly inhibits it in an allosteric site. Of course, you need ATP in its catalytic site to carry out the reaction, but it has a second site, which is a weak binding site for ATP, it inhibits it. But it has something even better. You really want to ramp up the rates of glycolysis. The way you do it is using an allosteric activator called fructose 2,6-bisphosphate. It's a different enzyme that makes essentially almost the same molecule, except the position is changed of the phosphate. That is the best known activator of PFK. It activates this enzyme 100-fold. That's the increase in activity you can get. You're doing numbers. AMP activates it by 3-fold, approximately 2-3-fold. If you have a large change in AMP, but you don't. Pyruvate activates it about 5-fold, and it's a good activator. But if you really need to ramp it up, you have this other molecule, which is almost the same as this, but it binds a different site, and that activates it 100-fold. So collectively, all put together, you can get 100 to 300-fold activation. Can you do this by just amounts of protein? Not easy. So transcription, making more protein is not going to reach these numbers, which means you can go essentially from a 0 to 100 using these modes of regulation. Yeah? Okay, we'll stop there. That was a good class. We're hungry. This is just showing all these allosteric activators bound to PFK and inhibitors. Everything there. So last slide, I'll leave you guys with this. So this was the thing that Jonathan actually ended his class with, movement of phosphates to release energy. You have high energy phosphate molecules and lower energy phosphate molecules, and the flow of phosphates is from higher to lower energy. So ATP is right here. This is why it can donate the phosphates to glucose and give you glucose 6-phosphates or something like that. But in order to make ATP, in other systems, you would use an even higher energy phosphate molecule. You have PEP. So that was just what happens in glycolysis here. The PEP can make ATP here, up here. You also have things like phosphocreatine in muscle and liver, which can also donate the phosphate to an adenosine diphosphate, ADP, to give you an ATP. Okay, so this is the continuous cycle. We'll stop. Any last questions before lunch?
If the activator that you just talked about, right, the two equivalent.
Oh, you mean here?
Yeah, so there. So if that is not a product of this pathway, then how does the regulation take
so
place?
that's actually incredibly beautiful biochemistry, because the most regulated metabolic enzyme we know is PFK. Like the second most regulated enzyme is the enzyme that makes fructose 2,6-bisphosphate. It's a different enzyme. It's a kinase phosphatase bifunctional enzyme. That is activated when you have sufficient fructose 6-phosphate, and it carries out a different reaction to make fructose 2,6-bisphosphate in very small quantities. Now, when we come back to these affinities and all of that, the affinity of this enzyme for fructose 2,6-bisphosphate is at least 1,000 times higher than its affinity for fructose 1,6-bisphosphate. So you need to make very small amounts of that to fully activate this enzyme, which will then carry out this reaction, and it won't be affected by this molecule. Yeah. So that's actually the whole textbook of how allostery works can be studied just by studying these two enzymes. Yeah. So why is it useful? Supposing you're doing glycolysis at a certain rate, the cell needs even more ATP. What do you need to do? You need to release the brakes of the reaction up here so that there's a flood of this coming in to make more of this. So that's why you'd use pyruvate over here. Make sense? Same with AMP or something like that. Way downstream, that will allow you to create a feed forward loop or an amplifying loop. If you have an inhibitor, like ATP, it's an inhibitor of this, that will give you a feedback loop to close your, reduce your reaction. Yeah, but because again, these reactions are happily going over here, they'll just be going. But if it's insufficient for the cell, this is what is giving you your payoff. You're getting ATP only here. You're getting ATP only here, and you're getting ATP. Yeah. Your payoff is coming here. So this is the net gain. This is all net zero over here. You're getting money. Your return on investment is coming here, no? So you're actually measuring return on investment. You actually don't in certain tissues. This enzyme is not present everywhere in high quantities. It's really present mainly in the liver and in the muscle, which physiologically, and in the brain, which are the three places where we're having maximum glycolysis. Yeah, where did that go? It doesn't decrease the Vmax, but it allows you to operate at Vmax. Yeah, but so for PFK, it's like this. Okay. Actually like this without the allosteric activator. Your concentration of, it's actually a pretty crappy enzyme without all the allostery. the concentration of these intermediates are in the low millimolar, and that's somewhere here. You need this allosteric activator to get you to Vmax in this concentration range. Concentration range is all relative. So even though glycolytic intermediates are all low millimolar, many of those enzymes have Kms at that range. So if the enzyme was operating at a much lower Km over here, then it doesn't need an allosteric. Okay, good. 2 more questions. That's 1 and 2. No, it's not. I mean, there are like tens of thousands of millions of phosphorus, equal opportunity. They're all there. Whichever one is there goes. Yeah. Yeah. Yeah, that's right. Not that I know of, but what we do have is situations. So, for example, many of the extremophiles in these hydrothermal vents have a bunch of highly unfavorable reactions present. Part of it is also for the biology, it needs to do those reactions to survive in those conditions. But clearly, they're operating at the edge of what is possible. Most of those are very, very slow growing organisms also. So there's some massive trade-off that happens between growth, biomass, and delta Gs. But, you know, if you're looking for life outside Earth or on all these places, we'll have to challenge how far we can go. We can't break the law of thermodynamics. Yeah, net will be negative, but we just have to find out where that net negative is coming from, because it won't come from a simple conventional ATP. Something else is doing that work. Yeah. So there is nice new biology to find, if in theory. Good. Okay. Lunch.